To begin, you would clone the repository. You would adjust the local host to 4,000 if you're running on a Mac, or keep it as 5,000 if you're running as a PC. You would next run views.py on your terminal. Once this gets running, you can now access the web app. For the sake of this video, I have created a sample DNA sequence. I would then download the sample DNA sequence. Once this has downloaded, I would go to Tools. I would select one of the 3 assembly standards. For this video, I would choose BioBricks. I would then select the file and then download the sequence with scars. You can then find the new sequence in the downloads folder. To make sure this is running, I am going to check the new file, the new sequence with scars. As you can see here, the scar has been added to the sequence that I have created here.
All right, welcome to this part of the demo where I'm going to be explaining how we read the XML files and then how we write the XML files into the user's computers. Now, to start off with, I want to say that we find the data that is important to us in order to understand the circuit in its totality. And what we find is the sequence of a given part. So like the nucleotide sequence, then the order of all the parts. And then we find the type of each part according to its name. So that means whether it's a promoter, terminator, etc. And then we are able to output the sequence, the type of the name, all in ordered sequences, or rather in ordered arrays, aka lists. Now, to do that, we read the XML file. We go into the important parts of the XML file. For example, to find the sequence, we find it in sequence. To find the order, we find it in the DNA's segments part, where this identifies the order for each part. And one of the main problems we found while trying to build this was that SBOLCanvas allows different kind of file type or like file arrangements, where iGEM, for example, has all these parts as well that are not in a normal SBOLCanvas output. However, we were able to generalize the code enough to where that was not an issue. Now, for actually writing the XML file, what we did was we created different formatable strings where each one just encodes for a different part in the XML file. And as expected, all we did then was simply format the strings, concatenate them together, and finally output a file that included all of the necessary information. It'll be named circuit with scars, 1, 2, etc. if one of them already exists. And that is the entirety of this part of the code.
Hello, I'm briefly going to touch a little bit on the biological background of our tool and some of the code that was used to actually implement the scars themselves. So firstly, I want to go over quickly the BioBricks standard, which is the first assembly standard that our tool supports. It's the most commonly used standard in DNA assembly within synthetic biology at the moment, and the majority of the iGEM registry of standard biological parts is compatible with the BioBricks format. Essentially, every part is flanked by the same restriction enzyme sequences on both sides, and you're able to cut these restriction enzyme sequences with restriction enzymes and piece them together in destination plasmids. And after we create a composite part, for this example on the left, we see that this example can also be put together with another simple part, as we see here on the right. So we see that this is a cascading process, and after every subsequent iteration of combining parts, they are flanked by the same sequences. So the larger and larger parts you get, you still have the same sequences, and you can keep repeating that process. Next, I want to briefly touch on the Type 2S assembly, which is a little more complex, but there's some fantastic information on the iGEM registry website on both these methods. The Type 2S has a little more flexibility in allowing the biologist to choose the overhang sequence exactly. And because of this, there's various substandards within it. The substandard that our tool supports is the Golden Gate modular cloning method, which has 3 levels of constructs. Level 1, as we see up here, is very similar to BioBricks. It's just the individual parts themselves. These are put together through various fusion sequences until you build up a transcriptional unit, which is seen here in level 1. And this transcriptional unit is essentially what we saw with the BioBricks standard. We have promoter, ribosome binding site, coding sequence, terminator, etc. And these transcriptional units can then also be cascaded and assembled together through another set of fusion sequences, depending on the length of the total part and its neighboring pieces. And constructing these into larger composite components gives us what is called a multi-gene construct. Quickly, I want to go over the implementation of the code of the addition of some of these scars. If we go over here, we can see that our code at the top just has an enumerated type, which allows us to check for the assembly standard that the user has chosen. Following that, we have our first class, which is to assemble 2 simple parts together. We begin with a constructor, which actually takes a series of arrays taken from the code that Jin and Antonio wrote. And these arrays consist of information that I'm sure, as they mentioned, has the part type, the part ID, and the length of these arrays. And through this, we're able to construct all the arrays that we're going to use to perform our analysis and addition of scars. The first step is we have a function which iteratively checks each part for illegal sequences within the part itself, not the restriction enzymes, because this would hinder the assembly process. So we iteratively go through and check based off the assembly standard that the user has chosen if certain illegal sequences exist within the sequences of the part themselves. And we do this for our other assembly standards as well. The next object function is the addition of the scars themselves, which again, through an iterative approach and the user's selection, we're able to incorporate the correct scar sequence between each parts. And we also check, depending if the next neighboring part starts with a specific sequence, there's another scar that is needed, which we found out through reading the literature. And we're also sure to add the correct, for example, in this case, BioBrick prefix and BioBrick suffix to ensure that these parts can once again be assembled with other composite parts to cascade this process. And this continues, same structure continues for our other assembly standards. And I want to briefly touch on one challenge that we had, which was actually putting together these already composite parts into larger composite parts, this cascading event that I've been mentioning. This was difficult to implement just because it was hard to take information from 2 different XML files and parse all the part IDs and the part types and be able to put them in an array, which would follow the same structure of code as I previously mentioned. So the code here is written very similar to the code above, and it functions if you manually input these arrays. However, we just had some challenging with that interface between the parsing of the XML files and incorporating them here. So this is one technical challenge that we faced that we weren't able to fully get incorporated, but code is right there, and this feature is could easily be added in the future.
Sure, after this video, I'm going to talk about how our front end and our back end communicate and then how it gets up to the web. Inside of our main.py, we call all of our functions. So inside of our views file, which is our main Flask app file, we're going to go down to the tools webpage, and then you're going to see that we have our imported main, which needs those 2 inputs to give us that output. So here's our tools page, and we're going to have the input for the assembly, and we're going to have our file, and then you can download that output file. We also wanted to take this from a local app and move it up to the actual web. So we do that through using a Docker file and a proc file. Docker allows you to run it locally as a Docker image, and then you can push that to a Docker registry. And then proc file takes from the Docker registry and then puts it up using a Heroku web service. So I'll show that over here. This is our local app. Here's our local Docker app. And then finally, here's our Heroku app for my beta sequence accurate SBOL application. As you see, these tabs are a little bit different than our final app, but this was a beta demo for the web app because we came to find out that the web app needs a paid database in order to communicate, in order for us to communicate with the user. So they can't get our downloaded file. It just won't go to their computer because of that paid database feature and security issues between local computers and the web. So in the future, we could pay for that and have our web app fully functioning, but until then, we will be running it locally. And yeah, that concludes this video. Thank you.